Hi there, it's Ms. Corcoran, and I believe I am starting with the chapter 24.1 and 25.1 PowerPoint about origin of life. You can see here, these are the learning objectives, and I'll let you go through them. This is copied and pasted straight from the College Board, what you are supposed to learn from these chapters. All right, what do we know about early life and early Earth? We know that the Earth is about 4.6 billion with a B, billion years old. And the early Earth, after the ball of rocks was formed, had an atmosphere that lacked oxygen. What was in it? Water vapor, nitrogen, carbon dioxide, methane, ammonia, hydrogen sulfide, a lot of really nasty stuff, but no oxygen. And there were all these volcanoes exploding and lightning flashing. And as that hot atmosphere cooled, water vapor condensed and filled in all of these gaps on the surface of this giant rock and made our oceans, which is kind of cool. The question that scientists don't really know, and they can only speculate because nobody was there when life began about 3.5 billion years ago, was did it form on the surface of the Earth? Did it form deep in the oceans? Did a meteorite slam into planet Earth and bring with it the building blocks of life? So you just need to be aware that there is one hypothesis, or a hypothesis, that sure enough, meteor brought these building blocks of life. In fact, the Murchison meteorite fell in Australia in 1969, and on it had monomers of organic molecules. So that's certainly a possibility. Did it begin on the surface of the Earth, or did it begin in deep sea vents? Let's talk about the surface of the Earth for a second. There are these fellas named Operin and Haldane, who in the 1920s said that the atmosphere was reducing, which means it adds electrons. And so as the atmosphere cooled and water collected on the surface, it was a reducing atmosphere, adding electrons, and that monomers of organic molecules formed and pooled in the oceans of the Earth. And they called it this primordial soup. The word primordial means like primitive and like oozy and gross and like primeval. So they hypothesized that back in 1920. And in 1950, two graduate students actually did this in a lab. They set up a scenario where, if you're looking at your slide, I don't know how I can do it. If you look at my cursor, I hope this is recording. In this ball right here, they set up an atmosphere like early Earth's. Methane, water vapor, nitrogen, everything but oxygen. And they zapped it with electricity to stimulate the lightning. And they had boiling water going up this tube, bringing the water vapor in here. And then there was a cooling tube, a condensing tube right here. And down here in the bottom, they collected some water. And within this water, they actually found the monomers to the building blocks of life. Remember from Bio 1, the building blocks, or the molecules essential for life, are proteins, nucleic acids, carbohydrates, and lipids. And really, proteins and nucleic acids are the big part of it. So I printed this out as a reminder from Bio 1 that a monomer is one unit of something. And a polymer is many monomers joined together. Mono means one, poly means many. And you learned in Bio 1 that, like, the monomer of a carbohydrate is glucose, the monomer of a protein is an amino acid, the monomer of a nucleic acid would be a nucleotide. So in the 1950s, Miller and Urey actually did this experiment right here, and they created the building blocks of life. Pretty darn cool. And it wasn't until like the 1980s that our technology had advanced to a certain degree in which scientists could go down to the seafloor. And they found some pretty remarkable things down there. I'm not going to spoil the video because you're going to watch the video. It's a Bill Nye, and who doesn't love Bill Nye? So I want you to watch that. But, you know, now people can hypothesize, well, maybe life began there on the seafloor. No one was here. We don't know. We can only speculate. But what we do know, and that has happened, is those monomers formed one way or another, whether on the surface or whether at the deep sea vents. And a scientist named Sidney Fox, and you don't need to know Sidney Fox, you do probably need to know Miller and Urey. But he actually dripped a solution of monomers onto some hot rocks, and he got them to spontaneously form together into polymers. So step one was getting the monomers to form. Check, we did that in the lab. Step two, can monomers spontaneously form into polymers? Yeah, Sidney Fox did that. Okay. What would be the next thing that would have to happen for life to form? Well, when you think about the smallest unit of life, it's a cell. And there's a membrane that surrounds every single cell. And the membrane separates the outside environment with the internal environment. And it can support a metabolism that is separate from what's going on outside for the most part. If you recall, a phospholipid, and we drew these in Bio 1 to look like this, sort of a balloon with two strings. This would be sort of the chemical representation of a phospholipid. But a phospholipid has a phosphate head and two fatty acid tails. And scientists have shown that if you dump a whole ton of phospholipids independently into a watery aqueous solution, it will spontaneously form a membrane. You have a couple questions on your Google Doc that I want you to answer, and you might need to look them up, but I want you to think about the properties of a phospholipid, the water-loving head, the water-fearing tails, hydrophobic, hydrophilic, and think about why that spontaneously happens. This video is only like a minute long, and it's kind of an animation that shows how it would happen, but it's totally spontaneous, and it happens every single time. So check, membranes have formed. Then there's this idea of a protobiont. Proto means like first, and bio means life. So protobiont is just this aggregation of organic molecules. Maybe it's nucleic acids, maybe it's proteins, but they're surrounded by a membrane, and there could have been some simple metabolism. So basically, and this is what you need to know, right here is the important slide. If life evolved, well, excuse me, it certainly did evolve. Life evolved on this planet, whether it was originated from meteorite, deep sea vents, on the surface. These 4 steps pretty much had to happen. We had to build the building blocks of life. We had to create polymers from monomers. We had to package them into these membrane-bound, like protobionts. And then we had to, self-replicating molecules had to form that carried the instructions for life. And now we know that to be DNA. So protocells were basically the first cells. They have been experimentally produced. So we've made monomers, we've made polymers, we've made protocells. The only thing that science hasn't done in a lab is get spontaneously to form these self-generating or self-replicating molecules of heredity. That brings us to this whole idea of the RNA world hypothesis. This hypothesis states that the very first living organisms used RNA as their molecule of instruction rather than DNA. Okay, this really isn't a stretch to think about that. There are 2 really big important supports here. Number 1, RNA does store genetic information. You know that from Bio 1 that the DNA holds instructions, but RNA copies the instructions and we can build a protein from it. So we know that RNA holds instructions. It's a code and it holds instructions. The other thing that's really kind of interesting about RNA is when we think about DNA holds instructions for building proteins, and proteins serve as enzymes which facilitate all of our metabolic reactions. Well, we didn't really have a ton of proteins then. Could RNA have served as an enzyme? You betcha. Because all enzymes do are make bonds or they break bonds. That's all really an enzyme does. So there's something called a ribozyme. It sort of sounds like a ribosome plus an enzyme together. And a ribozyme is RNA that holds code, number 1. And number 2, it actually facilitates chemical bonds. When you think about rRNA, which is the RNA that makes up the ribosome, at the ribosome, we are actually joining 2 amino acids together with a peptide bond. I'm throwing so many words back at you from Bio 1. So it facilitates the formation of the bond. So voila, a ribosome is actually a ribozyme. It is RNA that acts like an enzyme. Pretty cool. The last bit of proof that shows us that RNA could have been the very first molecule of heredity is that some viruses don't even have DNA, not at all. The only instructions that they have are RNA. So the fact that there are some pseudo-living organisms that have instructions as RNA tells us that it might have been the first molecule. Okay, so let's talk about evolutionary time. As time went on, DNA became the molecule of life instead of RNA. Yes, there are still some viruses that use RNA as the genetic molecule, but really it's DNA because it's more stable and it can be replicated more accurately because each strand, as it unzips, can serve as a template for making the new strand. Molecular evidence from all extinct and extant, which means currently living organisms, indicates that everything that's living on the planet share a common ancestor because every living thing uses DNA as its molecule of life. Now, I just mentioned viruses a minute ago. We don't really say a virus is a living thing. That's a whole different category, and we'll talk about that in January. So we threw out some dates earlier, 4.6 billion years ago, 3.5 billion years ago. How the heck do scientists know that the Earth is that old? Well, they use radiometric dating. They use known rates of decay of certain isotopes. And when they see how much of the parent material remains, they can determine how much time has passed based on known half-lives. So, for example, after 3 half-lives, a particular radioactive element has passed. How much of the original element is present? 3 half-lives. So let's see. It's easy for me to draw it kind of like a cookie. Here's the full amount of parent material. After 1 half-life, half is going to remain. So we have 1 half-life, half remains, okay? And then if that goes through another half-life, we only have half of the half. So a quarter remains. Then we're going to go through 1 more half-life, and we'll take a half of the quarter, which is going to be 1/8 of the original parent material. So I'm super visual. I have to draw things all the time. I hope that you understand that. If you don't, let me know. Okay, so let's talk about life. 4.6 billion years ago, the Earth formed right here. 3.5 billion years ago, prokaryotes, bacteria, unicellular, no nucleus organisms were first on the scene, heterotrophic and anaerobic, which means they had to consume food. They were consumers, and they were anaerobic because there was no oxygen in the atmosphere. It wasn't until much later that aerobic organisms appear because there is photosynthesis and oxygen in the atmosphere. If we take a look at this little clock here, we've got nothing going on, presumably, but really there was a lot going on. Prokaryotes, single-celled eukaryotes, and finally multicellular eukaryotes on the scene. A eukaryote has a nucleus and lots of membrane-bound organelles. So really for most of Earth's history, life was dominated by single-celled organisms. About 2.7 billion years ago, we know oxygen came on the scene, that photosynthesis was present because we can see oxygen in these iron bands in these rocks. And scientists are able to use that radiometric dating and determine about how long ago we started seeing oxygen banding in certain rocks. And the only way oxygen got present in the atmosphere was by photosynthesis. Okay, so how do we go from a single-celled, no-nucleus prokaryote to a eukaryote that's got lots of membrane-bound organelles? There's the idea of the endosymbiotic theory, that basically 2 prokaryotes were living close together. And this one said, hey, you come inside me and I won't digest you, and you're really good at making ATP, and I'll use that ATP. And so it takes the other prokaryote inside of it, and lo and behold, it becomes a mitochondria. The same thing happened with a chloroplast. Two prokaryotes are together, and the one says, hey, you come inside of me, I won't digest you. You're really, really good at making your own sugars. And it engulfed it and made its own sugars. And now you can't separate the mitochondria and the chloroplast from plant or animal cells and have them live on their own. It's impossible. That sounds pretty far-fetched, but there are three really important pieces of evidence that you need to know that support this. Number one, right up here, mitochondria and chloroplast have their own DNA that is separate from the nuclear DNA of the cell in which they are present. That's a pretty big fat clue that it used to be its own living organism. Number two, if they have DNA, which holds the instructions for building proteins, where do we build the proteins? At a ribosome. So they actually have their own ribosomes as well. Okay, that's not a leap. And lastly, they replicate independently of the host cell. So let's take, for example, you're starting a new exercise regimen. And the first time you work out, you are really, really sore in your quadriceps, your thighs. And after about 2 or 3 workouts, you're not sore anymore because your cells have been like, oh, dang, they're really serious about this workout. We need to start making more mitochondria because these cells are asking for more ATP since she's working out. So they're reproducing independently of the cell in which they live. Host cell probably isn't the right name for it, but the cell in which they live. All right, fast forward hundreds of thousands, millions of years, there's something called the Cambrian explosion, where there's a huge, rapid diversification. We see a lot of defensive armor. We see lots of claws. So if you ever hear about the Cambrian explosion, and this is just FYI, once life evolved, it started changing rapidly based on the environment. Okay, now let's talk about the history of continental drift. You've probably all heard of Pangaea. Pangaea is a supercontinent where everything was connected. That also sounds pretty darn far-fetched. I'm going to let you watch these 2 videos and see how the idea that this actually was a thing and how there is physical evidence supporting it. So you're going to do that on your Google. But basically, the Earth is a dynamic system. Life is dynamic. They are not static. It isn't like everything all of a sudden appeared and is never changing. The Earth is a constantly changing system. As a result, environments constantly change. And as environments change, they exert different environmental pressures on the living things that are present. And some will rise to the challenge because they've got the right adaptations. Others that don't, rip. Okay, that's the end of that. So I hope that this lecture helps you. I'm going to also post the PowerPoint so you can individually watch the videos and fill out your Google Doc sheet. Have a great day.